Okay, hello everyone and welcome for the second session of the Genome Application Symposium. This will be devoted to challenges in conservation genomics from analysis to essential biological variables. And I would like to introduce you, the chairs, Camila Mazzoni and Ivana. Thank you very much for accepting the challenge of leading this session, and I'm looking forward to the discussions that will be generated. Thank you.
Thanks a lot, Jose. Hello, everyone. Good morning. So I'm Camila Mazzoni, Group Leader in Conservation and Evolutionary Genomics at the Leibniz Institute for Zoo and Wildlife Research in Berlin. I'm here chairing this session together with Ivana. She will introduce herself in a minute. So I would like to tell you a little bit about how this session is going to be organized. So we are going to be talking about challenges for conservation genomics, first from the application point of view, management point of view, and Ivana is going to give you a presentation. After that, we're going to have the first discussion session, and I would like to invite you all to participate. When we arrived at the discussion points, that you can open your cameras, you can raise your hands, and throughout the whole session, you can put your questions or comments in the chat. So the idea is that we gather more experience beyond ours. So what we are presenting here is our own experience and our own understanding of the topic. And the idea is to spread this and to have as much participation as possible. After that, I will give an introduction to EBVs, and then we're going to have a second discussion at the end of the session. So Ivana, you can share your screen and introduce yourself.
Okay, hello everybody, good morning. It's a very nice Monday morning here in Zagreb, very sunny, and I'm very happy to be with you today. And in this first presentation, in my presentation, I will really focus on some of my experiences in how we now implement conservation genetics and conservation genomics, and what are the possibilities that are maybe still not completely used and maybe could be better used in the future. And I really hope you will help me here and that your opinions and your experiences, agreements, disagreements, etc. So in this symposium, we will start with challenges. Maybe that is even the best way when we are combining these two very young disciplines. And I hope you are seeing my slides. Can somebody please just wave or no? Okay, thanks. So that I know that everything is okay. So just to start, I have put here just a couple of definitions, maybe just to remind us what are we really talking about. For conservation biology, I have included this first definition that I like very much of Michael Solé, the father of the conservation biology, who coined this term in 1985. And genomics, actually, the term is only 1 year younger. So here we really deal with two very young disciplines, and are already combining them for some time now with more or less success. Maybe the first challenge comes from that these two disciplines are still mainly practiced by different people, different scientists. There are different departments in institutions dealing with them. They largely rely even on maybe quite distinct methodology. For example, in our faculty, and I will include here, this is really a nice symbol of our faculty. So I work at Faculty of Science in Zagreb, Croatia. This symbol that is really nicely illustrating how DNA investigations are important for biodiversity description. Completely different teachers are teaching these subjects, so it is quite distinct still. Anyway, we wouldn't be here today. We don't really recognize the link between these two areas that eventually led to the onset of this new discipline, conservation genomics, which is even younger. So it started sometimes in 2009 or something like that. So in this presentation, I will really present some of my opinions and my experiences and some of examples that I work on where we do or could use genetics and genomics for better practical conservation of some species. You may agree with me on some points or disagree or have some other experiences, but please, you can put them in chat so we can discuss them later, or later raise your hand and please share your experiences with us. So to start with, I have prepared a list of some parameters that are usually used in conservation of species, in real conservation, that are estimated to make conservation efficient. Some more often, some not as often. Not all of these parameters are used for all species. We do not have usual information for many of them when we try to make some conservation plans and some real conservation activities. Some of them are really essential for conservation, and Camila will later touch this much more in her presentation. Some are not as important, but in my opinion, this is the list of parameters that are or can be important for real practical conservation. I know many of you would add maybe some parameters here or put some out, but that I guess depends on our experience and our knowledge. You can, of course, challenge this in the chat, but I don't think this is so important to what I want to the point I want to make. To this list, I have applied two questions. So the first one is whether these parameters can be estimated using genetics, and even better using genomics, precisely and efficiently. And by this I mean cheaply and quickly enough, because all conservation projects have budgets, and these budgets are almost always not sufficient for everything we want to do. So I answered to those questions based on my knowledge and my experience. I put why if I think, yes, genetics and genomics is really important in estimation of that parameter, no, if other methodologies are more appropriate, and M, if it is a maybe or in some cases, or I'm not sure. So surely population size can be very precisely determined using genetic and genomic methodologies. Age structure of population, maybe for some species, but for many, no. So it's no. And sex structure, again, it can be for some species with sexual determination of sex. For others, it cannot. So it's M here. And I put my answers to all questions. And as you can see, for some parameters, genetics is important in estimation, in my opinion. Then I pose the second question, the red one. Is this parameter significant for implementation of the most relevant European documentations that tackle biodiversity conservation? So Habitats Directive, Bird Directive, European Strategy for Biodiversity, even extinction risk estimation, etc. And again, I answered whether these parameters are used, are covered by any of those documents, the most relevant ones. So here are again answers. I really acknowledge that maybe from experiences of some of you, you will have different answers, and think that genomics is maybe more important for some parameters that I prefer to estimate using other methodologies. But that is really not so important for what I want to show here, because it is very visible that we do have many parameters that can be efficiently and precisely estimated using genomics, and are important for implementation of these relevant biodiversity European documents. That does not mean that these parameters are currently widely really estimated using genetics or genomics. I will come to that in a minute, but they can be really correctly estimated using genomics. There are also a couple of parameters that I find, from my experience, important for practical conservation of some species, and that genomics is important for their evaluation, but are not covered by these European documents. So here we have like maybe two discrepancies. I will now try to go in more details in the application of both genomics and European legislation on one example. And the example is, of course, this small endemic fish that is one of the fishes that we focus on in our case study under BGE. So it is Orsinigobius croaticus, Neretva dwarf goby, very restricted distribution range. We find it highly endangered. So I will just run you through how some of these documents were applied concretely on this species. So we will start with IUCN Red List, so extinction risk estimation. Yes, for this species it has been conducted, and it is considered to be vulnerable, so facing high extinction risk. However, its extinction risk estimation was based only on its distribution range and the quality of habitats. No other parameters have been used, so only criteria B and D of these 5 criteria that IUCN uses have been exploited. And here I can see some problems. If you can see some others or disagree with me, please use chat and tell me what you think. First, these all gobies are morphologically very similar, and their taxonomy is not very well settled. So from that, it comes that the number of locations is questionable, because some population that was determined as Orsinigobius croaticus might be completely different species, but only morphologically similar, or some populations of Orsinigobius croaticus might be neglected in the estimation. And number of location makes very much difference when we use this criterion B. So it's very different if the number of location is 1, 6, or 11. That makes difference between critically endangered and nearly threatened species. So this is the first problem. The second problem that I see is that no other parameters are included, and maybe even very widespread species can have some internal genetic problems that are really making them facing extremely high risk of extinction, which is neglected. So in my view, genomics provides us here with some quite nice possibilities. And we do hope to fulfill some of them during this project for these concrete species. By correct identification of taxonomic units, of course, we can really have precise information of the area of occupancy, so on distribution range and number of localities. But my opinion, and it has been my opinion for some time now, is that in addition to these 5 criteria that are the basis of the extinction risk estimation under IUCN, I would really like to see in maybe recent years addition of 1 or 2 more that are based solely on some genetic important variables that do determine extinction of the species. Maybe I'm dreaming, but I think that would be very useful. Then the next document that can be applied is Habitats Directive. So Orsinigobius croaticus is listed on the Annex 2 of the Habitats Directive, and based on that, Natura 2000 sites have been designed and are managed with the aim of protecting these species. So here again, I see some problems. Again, if we do not really well have recognized the populations, then it is possible that we are managing some Natura 2000 sites that are occupied with other gobies, not this species, and that some localities are not covered, which is problematic. And again, for this species, conservation actions, they did not consider any genetic or genomic results, so no genetic structure, diversity, or such parameters that are actually important for the viability of species have been included. They can be included if the knowledge exists, and we have them in management plans for some species, but here not. Anyway, possibilities are again great, not only in this determination of distribution range, but also for really understanding the structure of the species, identifying maybe source populations, so the ones that are more important for protection, or source population for introduction, augmentation, etc., which is also something that could be even added maybe in this important documentation. I mean, Habitats Directive relies only on species, and this is something that we could discuss maybe on some other symposia, and it will take us some time to share this consensus, but I don't think conservation of species is good enough. And there are many evidences that we should conserve units below species level, and that would be maybe more efficient way to protect biodiversity. Then practical conservation for this species, it relies on it being Natura 2000 species, and I already, I think, covered most already. So here are the activities that are really in the management plan of this species. Again, the problem is that no measures are targeting any internal population parameters, only some general conservation activities, which is, well, from conservation practice, we know not efficient enough. Possibilities, again, high, not only in preserving and designing efficient conservation measures for this and other species, but also, for example, for early detection of invasive alien species, and very, very nice tools for monitoring that are, again, not as exploited as could be. I will just run through some other documentations, maybe just also to have them in mind and to maybe, well, shortly discuss whether genomics can even aid their implementation. So EU biodiversity strategy, it does really nicely target the protection of all European biodiversity. But here again, genetic diversity is not really accentuated. It is really focusing on species, which is okay. I mean, if we would protect all European species, we will be very happy. But is this the most efficient way? I don't think so. I think genetics and genomics can really be useful here, and they maybe should be already included in strategies. Regarding EU Water Framework Directive, at this point, my opinion is that classification systems as they are now are very efficient. But again, there is a room to discussion, maybe how eDNA analysis, and then we again come to genetics and genomics, can also aid to this. So to conclude, again, my opinion is that really genomic could have really high application in practical conservation, so can be really closely linked to this area. Not always, not in all cases. It doesn't have to be forced. It shouldn't be forced, but those possibilities should be investigated more. And as a proof for that, my opinion, I can say, again, from my experience, that in cases where we managed to include some conservation genetics in real conservation programs, it was really successful, and it really helped conserving some species. So I will conclude with this nice quote, and with the video that was filmed by my young colleague, Nikola Renic, who you see here, and who shows two things. First, how for now, combination of conservation genetics and conservation practice, and we do that on Plitvice lakes, in managing endangered trout species, where we really for spawning investigate genetics of the pairs that we spawn in order to introduce really hatchlings that do have genetic constitution that is important for a certain area. And genomics would here be much better because we would have an overview of the whole genome. And the second thing is how by maybe putting such short videos on social networks, what my young colleagues do, how can we spread the word on this usefulness and benefits on genomics and make it maybe more challenging for our stakeholders. We'll talk more about this tomorrow, but it is again an important point. So I will just show this small video to finish. It is short. It shows this the hatchling site where we check the individuals that are used for spawning and make spawning of adequate phase and bring the eggs after they have gone the most sensitive phase into the localities where they are necessary to raise effective population size and genetic diversity of populations. Okay, they're put now in the water, and with that, I will thank you very much for your attention, and I will stop sharing and ask Camila whether she was maybe checking the chat during my presentation, and if we do have maybe some questions, or we have some hands raised.
Thanks, Ivana. Yeah, I will ask. So if you guys have questions for Ivana, feel free now to raise your hands. I'm also going to share the screen here with a couple of points that we thought could raise discussions. So if you want to add to any of this in the chat or out loud, or also in your heads for conversations later. So we are talking here about the challenges and perspectives for the use of genomic methods for conservation. So we want to talk also about the limitations in the application of the current genomic technology. So Ivana has presented a good couple of examples where genomics or genetics can be used or to improve what is out there, our estimations and perspectives. But what are the limitations for that? Also, which methodologies are best suited? Because genomics is a technology that is quite broad, quite variable in the types of applications. So are there methodologies that will be more suited for direct application and conservation? And also, how can genomic data help predicting future biodiversity trends? So we are talking a lot about the assessment now. How will genomic help us in the future? And how does this differ from all the other type of data that is out there? And finally, how can we improve the accuracy of the genomic data that we produce, so the analysis that we produce for conservation purposes, maybe with more targeted approaches, or maybe with some other more generic approaches that will increase this accuracy? So we're not going to have a lot of time to discuss because we need to go to the second part of the session. So I will just open if anyone has one comment. Otherwise, I will ask even Ivana to try to answer one of these questions. Anyone has anything to say?
I don't see any hands. I guess maybe the first coffee is not finished yet. Yes, I have mine here. So cheers to everybody. Yes, I think I have said my opinion to most of these questions actually during the presentation. But again, I can maybe focus on these challenges with which I start. Is that okay?
Sure. Yeah.
Maybe summarize what I find are the greatest challenges in my opinion. But feel free to jump in and also share your experiences. So as I said, firstly, it is still Maybe even only in our heads, quite distinct. I mean, persons who work in labs and are very, very skilled in genomics sometimes are not so connected with conservation practice. It's different, and it's really, really distinct to work both. Because in conservation practice, you have to be in the field, you have to work with the species, you have to work with stakeholders. You don't have so much time really to go deeply into this methodology. So it is distinct. I don't think that's something that cannot be bridged, of course. I mean, multidisciplinary teams are necessary for some real conservation, so they can have somebody really skilled in genomics and who can interpret results well, and also some conservational practitioners. But I can define that as one challenge. The second one that I see from my perspective is that well, still in Europe, conservation is very slow. I'm sorry. I don't know how to better to say it, but I don't know, Habitats Directive is from 1992 and it didn't change. And so even newly described species, it is so hard to put them there. IUCN criteria have never been challenged or changed. And maybe the time came really to change that. And then maybe the third thing is that we are lacking discussions and symposias and meetings and collaborations like this one. And in that sense, I really see the BGE and even this symposium, but the whole project really important for maybe solving some of these challenges and putting really genomics aiding conservation. So that's my opinion. I see some hands, so
I will just ask for you both to be brief if you can. So let's start with Leif.
Yeah, so I mean, I fully agree with you that in order to apply genomics, you need to study populations. Then you need to do population studies, which require a lot of time to collect samples and that. So, and I also would like to raise one thing, and that is that it's also important. We often talk about rare species and species at risk of extinction, but I think it's also really important to study common species which contribute to the ecosystem. And I will give an example tomorrow from Atlantic herring, where we recently discovered a new ecotype of herring that was completely unknown before, which would, if you just were using barcoding or something, you will never see this. You need to understand the population structure to see this. So I think that is also very important.
I fully agree. Yes, thank you. Jose also has a question.
Yes, just very briefly. So one very important aspect of promoting application is to generate consensus on what are the measures, the parameters that are relevant for application and we end up in the essential biological variables that I believe Camila will talk in a minute. And for that, what I feel is really important is standardization, because if we first agree on a set of parameters that are the standard ones that can be included in policy frameworks and things like that, the next step is then to standardize analysis to develop simple toolkits that can mitigate a little bit that problem of avoiding that everyone would need to be an expert from bioinformatics to stakeholder to being a practitioner and bioinformatics in the same person. So I think standardization of and the agreement on a set of parameters that are the basic ones that are informative about natural populations is really important to
Yes, also. Yes, I agree. And we have a short question in the chat by Joao. So what genetic genomic points might be the most important to include in conservation criteria? Well, again, from my experience, although this is something that expert group should discuss and maybe apply to some examples and then see, I think genetic diversity, because it is easy to investigate, it can be just a single number. So it can be very well used and applied for criteria like other criteria are. And on the other hand, it does represent everything that has happened to these species in the past, whether they have gone through some bottlenecks, so it is lowered and special care should be taken, and that makes some risk to species. So that will be the first candidate in my opinion.
Great,
So now,
thanks, Ivana. Yes, I will continue.
I wanted to give floor to you anyway, don't worry, and thanks everybody for participating.
Thanks a lot. And please continue in the chat. If you want to continue this topic, just continue in the chat. Ivana and the others can go there. I will actually now hopefully wake up most of you. So break a little bit the ice. I would like to ask you, so we're going to talk a little about EBVs. It's just going to be a brief introduction, but I want to know how much you actually know about EBVs without Googling, please. So please go to this web, either use the QR code here if you have a phone, or go to this webpage, menti.com, and use this code that is over here. Okay, if someone can put this code in the chat, I would appreciate. And there you're going to answer whether you have good knowledge or have no idea. And this is all anonymous. So we're going to have an idea about our audience here today. How is this distribution? Okay. It's good to know that there are a few people with very good knowledge, but I already expected that many would have never have heard of this term even. Nice. So I'll give you another 10, 15 seconds or so. Okay, it's increasing. Yeah, so I think the trend that we have is probably not going to change. The great majority hasn't heard or maybe heard, but have little clue about what they are. And we're going to mention these EBVs now because this is going to be very important in policy in the coming years. And we, as biodiversity researchers of any kind, we should know about them and we should think about them. Can we do something? Can we participate? So this one is the never heard of EBVs, and as you see, is the great majority. Okay, thank you so much for the inputs. 61 people gave the input, so that's very nice. And I will go back to the presentation now. So please let me know if you can see. Can you see now my slides presentation? Yes. Okay. So let's give a brief introduction to this term, the EBVs. Now, the EBVs, so before we even get into the EBVs, let's talk about what are we living right now. So since the last COP15 that happened 2 years ago, so in a month or less than a month time, we're going to have the COP16, this new framework, the Global Biodiversity Framework, was agreed upon with a set of targets that the countries need to reach and have action towards these targets until 2030. So very short term when we think about policy changes. There are, as I mentioned, 23 targets, and if you go through them, you will see that several of them do connect with genomics somehow, or even potentially. Yeah, some directly. So when you talk about access and benefit sharing of genetic resources, digital sequence information, this connects directly. And some others, we as the genomics community can very well imagine genomics being applied and helping achieving those targets. But maybe for the sake of today, talking about conservation, target 4 is the one that is applies the best for that, where the aim is to halt human-induced extinction and recover and conserve species. So especially threatened species, so the focus is there, but not only. The important thing here is that there is finally a focus on wild species, which was not there in the past, okay? Maintain and restore the genetic diversity within and between populations. So this aspect of looking at the populations within species is now and highlighted and important as well, and maintaining their adaptive potential, which is maybe the most challenging part of this target if you think about how one can assess this. Yeah, but there are ways that have been developed, at least to try to do this indirectly. So the essential biodiversity variables, now these EBVs in capital, of course, many of us can have our own biodiversity variables and some of them will be essential for our specific cases. But these ones have a special meaning. They were developed by this group on Earth Observation, Biodiversity Observation Network. So it's GeoBon. So back in 2013, that was before the GBF, so this Global Biodiversity Framework. It was when there was the Aichi targets that were set for 2020 that were not that successful as they should have been. So this group has come up with these essential biodiversity variables inspired by all the variables, the essential climate variables that help in the implementation of the global climate observing system. So these variables, the ECVs, are apparently very useful and very successful in helping in the implementation and the actions against the problems that climate change bring to us. So these essential variables, and thinking about the essential part of it, is that there should be a set of a minimal set of variables that will capture biodiversity complexity. So you can see the trouble there, that we are talking about enormous complexity, and now we are trying to find a set of variables that will capture that. And this is with the goal that these variables will help us study, report, and manage biodiversity change. So this, the EBVs then are subdivided into 6 different classes. 3 of them are focused on species and 3 of them are focused on ecosystems. We are going to direct our attention here to the species-focused ones, where we have genetic composition, species and populations, and species traits. So the idea then is not that we're not talking about a species, one management program in one place, but it all starts with this. It all starts with the local scale, such as the example that Ivana gave to you today. But how to do this now at a level where you can jump from local scale to the transnational scale and finally end up with general trends of indicators about what our actions are doing in terms of the biodiversity change. So how are we improving or not the conservation and restoration of biodiversity? So these essential variables then, they represent an intermediate level or data layer for harmonization between sampling protocols and measurement systems. So whatever the raw data that we generate at a local scale with the analysis, all the way to general trends and indicators that the politicians, the policymakers understand. Okay, so then this is the idea in reducing the number of variables, but is still capturing as much as possible of the information, the complexity that is needed, and that is going to inform us about the changes. So these variables, they should inform us about changes across time, across taxa, and across regions, and even types of ecosystems such as marine, freshwater, terrestrial. So going more specifically now about the genetic composition EBVs, also the GeoBon group has come up with a set of the essential biodiversity variables, 4 of them more specifically, namely the genetic diversity, genetic differentiation, inbreeding, and effective population size. with the idea that in combination, this will help us understand what is happening in terms of the internal variability of populations. So are they being kept? Are they being lost? Also, the populations that exist, how are they structured? And here, if you remember the adaptation point that I brought up before in target 4, this is the indirect way of looking at, of guaranteeing that adaptation that does, or adaptive features that exist within species are not lost by keeping the variability across populations. This would also keep the adaptive capacity of the different species. So it's an indirect way of guaranteeing that. Levels of inbreeding, of course, because as more related individuals start generating offspring, this is going to decrease rather quickly the genetic diversity. And the same for the effective population size, where actually this is this parameter here is giving a even broader attention, because this was even turned into an indicator. So all the way, not only EBV, but all the way at the end, where the policymakers are looking into this. So this is now, I think you have seen all the list of parameters that Ivana listed, and where she is putting their emphasis when thinking about this one species of fish that is vulnerable and that has a management program. But then how to do this at scale, how to do this over time and across taxa. And one of the things that the genomic community can look into is, from one side, how can genomics help informing this at not only in a precise way, but also in a practical way. Because as we said, this is not going to be scientific studies. This needs to be in practice and in the field. And on the other hand, are there other variables, essential variables that this community would think are important to be tracked over time? So with this, I leave a few points. We only have a couple of minutes, so if anyone has anything to contribute, I'll be super happy. So the few points for discussion in that would be the challenges in linking genomic data to EBVs, as I mentioned, how to ensure that EBVs are relevant and useful for conservation efforts. So not only that they inform the trends, but they actually inform the efforts themselves. Genomic data informing policy decisions. How can we make this happen? What are the challenges in maintaining genetic diversity in conservation program? How can conservation genomics be made more accessible to developing countries? So even in some management programs from developed countries, this genomics is maybe already feasible. How to make this feasible for developing countries as well? And what roles do stakeholders, so the ones that are in the field or directly managing the programs, are playing in the generation and the use of EBVs? So these are points that, again, we're going to leave out there. We don't have much time, but if anyone has something to contribute now, just as a last contribution to the session, I'll be very happy. So
we do have just one observation in the chat, Camila, if I can interrupt.
yes.
It seems that some people are maybe familiar with essential biodiversity variables, but didn't recognize the short. So maybe the trend would be a little different, but I think this introduction was really nice for many people, and thank you for that. And
but
Camila,
that was the point.
Yes.
That was the point, because EBVs, they're not spelling it out. They use now from COP and from policies EBVs. So that's what I wanted to know, if people knew what EBVs were.
Yes, that's right. It's also the shortening for virus.
Well, I'm sure there are other meanings for that. Okay, Gabor is saying something here. All these genetic characteristics of species that compose the genetic element of EBVs are exclusively based on neutral genomic variation, or this is not defined? That's a really good question. Well, I'm not exactly, maybe if someone in this audience here was involved in coming up with this EBVs, I will invite you to raise your hand. But otherwise, no, I don't think they involved non-neutral genomic variation. To be fair, I don't think they even involved genomic variation. I think the idea there is that a lot of this, I mean, of course, the genetic, the heterozygosity and so on comes from genomic variation, or at least from markers. So the idea, I think, from the group was to gather what is already there, what is already achievable by the community, no matter what the markers were. And the majority of the markers that were used or have been used historically are neutral, such as microsatellites and others. Right, but even like as this abstraction or extrapolation of the effective population size, the use of genetic information is not even needed, at least following the current understanding. Yeah, so therefore, if we want to go even deeper into that, into the adaptive components of the genomic of the genomes, of course, this would take a whole new layer. That is something that we probably need to discuss, yeah? Because first of all, is it really needed? And second, is it feasible? And when will this be feasible? Because maybe it's not feasible now, but will be feasible later on. José, you have a...
Yes, I think there are actually two sets of challenge. First, the scientific community as experts need to agree what are really the minimum or the basic EBVs that can really describe a population and that are applicable. And then the second part is how to estimate these EBVs. And coming again to standardization, what are the methods or the basic methods to estimate each EBV? And then I think Katja makes also an excellent point, is that we need to see this, which she mentions, and I'll read it. There are processes as partially clonal reproduction whose effects on genetic variation and adaptability are not yet fully understood. Will it be possible to update EBVs as methods improve? And I think that's also an important aspect, is that we need to see this as a dynamic process.
That is the idea, that's what it's claimed, okay? One problem that I see potentially is that we end up more, or let's say, the EBVs or the developed of EBVs of indicators end up being much more influenced by the policy side than from the scientific side. And of course, there are obvious reasons for that, because it's only the policy side that can do something. But the question is, how can we, the scientific community, really have a say in there? So I think that's the big question that we should be discussing next. Gabor, I'll give the word to you, and then I will finish this session.
So thank you. Just a final remark on neutral variation versus adaptive variation. I think it's very important to emphasize what we pick from the genome, because sometimes adaptive variation is light years away. I mean, the diversity and the genetic structure is light years away from neutral genetic variation. So if genomics are included, then definitely I think it's important to define all these genetic characteristics to be measured from the neutral genomic variation. Thank you.
Thanks, Gabor.
Thanks.
Okay, well, that was short. José, I think we can close this session now so that we can have the break before the next
Yes,
one.
I think there are some aspects of this that we will discuss along the symposium. So I think we will come back to some of these issues. So thank you, Camila and Ivana, for driving this discussion. So we will now have a short break. So at 10:15 CEST, mind the time zones, we will come back with the Genomics of Adaptation session. So we will break now and be back then. Thank you. Thank you, everyone. It was very interesting.